Origin and Evolution of Life, circa 3.8 billion to 538.8 million years ago. Section 1. A planet ready for life. Before forests, before animals. Before eyes, bones, shells, teeth, wings, blood and breath. Before the first fish moved through the sea. Before the first creature crawled across the land. Before humanity, before civilization, before thought, before history. Earth was young. It had survived fire. It had survived impact. It had survived the violence of its own birth. The planet had formed from dust, rock, metal, gravity, and collision. Its core had separated. Its mantle had taken shape. Its surface had cooled enough for crust to appear. Water had begun interacting with rock. Volcanoes released gases into the atmosphere. Impacts still struck from space. Lightning flashed through ancient skies. Heat rose from the interior. Minerals formed along seafloors, shorelines, volcanic ponds, and hydrothermal systems. Earth was not yet alive, but it was becoming a world where life could begin. Its oceans were dark. Its atmosphere held almost no free oxygen. There was no ozone shield like the one that protects Earth today. The surface was exposed to harsh ultraviolet radiation. The planet was unstable, violent, and chemically active. But that chemistry mattered. Water could dissolve and move materials. Volcanic gases could feed reactions. Minerals could concentrate molecules. Hydrothermal systems could provide energy. Wet and dry cycles could force chemicals together. Impacts could destroy, but they could also deliver carbon, water, nitrogen, and organic compounds. Earth was no longer just a planet of rock. It was a laboratory, a world where chemistry could become more complex, a world where matter could begin organizing itself in new ways. And somewhere in this ancient Earth, across environments now lost to time, the boundary between non-living chemistry and biology began to blur. No one knows the exact moment life began. There is no fossil of the first living cell. No preserved record of the first self-replicating molecule. No ancient stone tablet marking the birth of biology. The earliest life was microscopic, fragile, soft, easily destroyed by heat, pressure, erosion, and time. Earth's oldest rocks have been altered, buried, melted, and recycled. The evidence is incomplete. But the search remains one of the greatest scientific quests in human history. How did chemistry become life? Section 2. The search for life's origin. The origin of life was not the same thing as the first fossil, and the first fossil was not necessarily the first organism. Scientists separate three great questions. First, the origin of life. How non-living chemistry became self-sustaining biology. Second, the earliest fossil evidence, the oldest surviving signs that life existed. Third, Luca, the last universal common ancestor of all living organisms today. Luca was not the first living thing. It was the ancestral population from which modern bacteria, archaea, and eukaryotes inherited their shared biological machinery. Before Luca, there may have been many experiments in life. Some may have failed. Some may have gone extinct. Some may have left no descendants. Some may have merged, exchanged genes, or transformed into new forms. The beginning of life was not a clean line. It was likely a long transition. Chemistry became networks. Networks became self-maintaining systems. Some molecules stored information, some catalyzed reactions, some formed membranes, some captured energy, some copied themselves imperfectly. And imperfect copying allowed variation. Variation allowed selection. Selection allowed evolution. This was the beginning of biology. Long before Charles Darwin described natural selection in living organisms, natural processes may have already been sought in chemical systems on the early Earth. Some molecules lasted longer. Some reactions repeated more effectively. Some systems grew. Some broke apart. Some became enclosed in primitive membranes. Some passed their structure forward. Life may have begun not as a single perfect cell, but as a messy population of chemical systems gradually becoming more organized, more stable, and more capable of evolution. Alexander Oparin and J.B.S. Haldane were among the first major thinkers to argue that life could emerge through gradual chemical evolution. They imagined early Earth as a place where simple chemicals could build into more complex organic molecules. In 1953, Stanley Miller and Harold Urey tested part of this idea. They passed electrical energy through a mixture of gases meant to represent early Earth conditions. The experiment did not create life. It did not perfectly recreate Earth's ancient atmosphere, but it showed something powerful. Organic molecules, including amino acids, could form through natural chemistry. Life's building blocks did not have to arrive already alive. They could be made. Later, other researchers expanded the search. Joan Orrow showed that important biological compounds could form from simple chemicals. Leslie Orgel helped develop ideas about early genetic molecules. Thomas Seck and Sidney Altman discovered catalytic RNA, showing that RNA could do more than carry information. It could also perform chemical work. That discovery strengthened the RNA world hypothesis, the idea that early life may have used RNA-like molecules before modern DNA and proteins dominated biology. RNA is remarkable because it can store information and catalyze reactions. It can act as both instruction and tool. But the RNA world still has mysteries. How did long, stable RNA molecules form naturally? How did they copy themselves? How did they survive harsh conditions? How did they become enclosed in early cells? The answer may not lie in one place. Life may not have started from one simple pathway. It may have emerged from the interaction of many environments: hydrothermal vents, volcanic ponds, mineral surfaces, wet and dry shorelines, ice, impacts, atmospheric chemistry, primitive membranes. Researchers such as Michael Russell and William Martin have argued that alkaline hydrothermal vents could have provided chemical gradients, mineral compartments, and continuous energy. David Deamer and Jack Sostak have studied how primitive membranes and protocells could have formed, grown, divided, and carried chemical systems inside them. John Sutherland and others have explored pathways by which biological building blocks could form under plausible early Earth conditions. No single origin of life model has been proven, but all of them point toward the same profound idea. Life was not magic dropped onto Earth from outside nature. Life emerged through matter, energy, chemistry, environment, and time. Section 3. The earliest evidence of life. The farther back scientists look, the harder the evidence becomes to read. A shape in ancient rock may look biological, but geology can imitate life. Minerals can form patterns. Heat and pressure can deform structures. Chemical reactions can create signatures that resemble biology. This makes the earliest evidence controversial. Some rocks from Greenland, around 3.7 billion years old, were once interpreted as ancient stromatolites, layered structures built by microbial communities. Later analysis suggested that geological deformation could have produced their shapes. The debate revealed how careful scientists must be. The oldest life cannot be accepted because a rock looks interesting. It must survive intense testing. More convincing evidence comes from the Dresser Formation in Western Australia, dating to around 3.48 billion years ago. There, scientists have found stromatolite-like structures, microbial textures, chemical signals, and evidence of ancient hot spring environments. These rocks suggest that by at least 3.48 billion years ago, Earth supported organized microbial ecosystems, not animals, not plants, not fungi, not complex cells with nuclei, microbes, small, ancient, powerful forms of life. The first ecosystems were microbial worlds. They may have lived in shallow waters, hydrothermal settings, hot springs, sediments, or mineral-rich environments. They formed mats. They interacted with rock. They used chemical energy. Some may have captured sunlight. They changed their environment slowly, layer by layer, generation by generation. Calling them simple can be misleading. They were simple in structure compared with animals or plants, but their chemistry may have been sophisticated. Even the earliest life had to solve hard problems. It had to capture energy. It had to maintain itself. It had to reproduce. It had to protect its chemistry. It had to evolve. The first life did not build cities or skeletons. It built metabolism. It built inheritance. It built survival. And from those microscopic beginnings, every later branch of life would eventually grow. Section 4. Life learns to use sunlight. At some point in Earth's early history, life found a new source of power. Sunlight. Before photosynthesis, organisms depended on chemical energy from their surroundings. But sunlight poured onto Earth every day, an enormous energy source waiting above the surface. The first photosynthesis was probably not the oxygen-producing kind that plants use today. It was likely an oxygenic photosynthesis. This means early photosynthetic organisms captured sunlight but did not release oxygen. Instead of splitting water, they used other electron sources such as hydrogen, hydrogen sulfide, dissolved iron, or other reduced compounds. By around 3.42 billion years ago, microbial communities were probably using sunlight in this way. They were not yet filling the atmosphere with oxygen. They were learning to turn light into life. This was a major turning point. Life was no longer limited only to chemical energy from rocks, vents, and local environments. It could begin harvesting energy from the sun. The surface of Earth became more biologically powerful. Microbial mats spread through shallow environments. Communities layered themselves across sediments and mineral surfaces. Sunlight became part of Earth's living machinery. But the greatest transformation had not happened yet. That came when some microbes evolved oxygenic photosynthesis. Oxygenic photosynthesis used water as an electron source. It split water molecules. It used sunlight to build organic matter from carbon dioxide, and it released oxygen as a waste product. To life today, oxygen feels essential, but to early Earth, oxygen was dangerous, reactive, corrosive, toxic to many organisms that evolved in an oxygen-free world. At first, oxygen did not build up in the atmosphere. It reacted with the planet. It combined with dissolved iron in seawater. It reacted with reduced sulfur compounds. It reacted with volcanic gases. It reacted with minerals in Earth's crust. For hundreds of millions of years, oxygen was produced locally, but the planet absorbed it. The oceans and rocks acted like oxygen sinks. Cyanobacteria and other oxygen-producing organisms were changing the world, but slowly. The atmosphere did not transform overnight. It had to be overwhelmed. Section 5. The Oxygen Revolution. Before oxygen could reshape the sky, it first reshaped the oceans. As oxygen entered seawater, it reacted with dissolved iron. Iron that had once remained dissolved began forming iron oxides. These iron-rich minerals settled in layers across ancient sea floors. Over time, they helped create banded iron formations, vast geological records of oxygen reacting with Earth's chemistry. The rocks became archives of a planetary transformation. The rise of oxygen was not just a biological event, it was geological, chemical, atmospheric, planetary. For a long time, oxygen production and oxygen destruction stayed in tension. Microbes released oxygen. Iron, sulfur, volcanic gases, organic matter, and crustal minerals consumed it. But eventually the balance changed. The sinks weakened, the oxygen remained. Beginning around 2.43 billion years ago, Earth entered one of the most important transitions in its history, the Great Oxygenation Event. This was not a single moment, not one day, not one year, not one simple switch from an oxygen-free world to an oxygen-rich one. It was a prolonged, unstable transformation lasting hundreds of millions of years. Oxygen rose. It fluctuated. It transformed oceans. It changed minerals. It altered atmospheric chemistry. It opened new biological possibilities. And it created a crisis for many organisms. For life that had evolved in an oxygen-free world, oxygen could be poison. It damaged molecules. It attacked cells. It forced anaerobic organisms into oxygen-poor refuges, sediments, deep soils, isolated waters, and protected environments where oxygen could not easily penetrate. Some life retreated, some adapted, some evolved defenses. Some learned to use oxygen, and that changed everything, because oxygen made aerobic respiration possible. Aerobic respiration could extract far more usable energy from organic molecules than many anaerobic pathways. More energy meant new possibilities, larger cells, more active metabolisms, more complex biological systems. The atmosphere also began forming an ozone layer as oxygen chemistry changed the upper air. Ozone reduced harmful ultraviolet radiation reaching the surface. Earth was becoming more protected, more chemically complex, more capable of supporting future life. But complex organisms did not immediately appear. For much of the Proterozoic eon, oxygen remained far below modern levels. More than 1 billion years would pass before large animals became common. Still, the Great Oxygenation Event was one of the greatest revolutions in Earth history. Life had changed the planet, and the changed planet would change life. Section 6. The Rise of Complex Cells. For billions of years, life on Earth was mostly microbial. Bacteria and archaea dominated the biosphere. They were small, ancient, adaptable, and metabolically powerful. But eventually, a new kind of cell appeared. The eukaryotic cell. Eukaryotic cells were different. They had a nucleus containing chromosomes. They had internal membranes. They had a cytoskeleton. They had compartments, and most importantly, they had mitochondria. Mitochondria allowed cells to process energy in powerful new ways. The origin of eukaryotic cells was not a simple upgrade. It was a merger, a deep biological partnership. An archaeal-related host cell entered into a stable relationship with a bacterium. Instead of digesting it, the host retained it. The bacterium supplied energy. Over time, it became the mitochondrion. Later, in the ancestors of algae and plants, another partnership occurred. A eukaryotic cell incorporated a photosynthetic cyanobacterium. That cyanobacterium became the chloroplast, This is endosymbiosis, one cell living inside another, a partnership so deep that separate organisms became parts of one new organism. Mitochondria and chloroplasts still carry evidence of this origin. They have their own DNA. Their DNA resembles bacterial DNA. They divide like bacteria. They have bacterial-type ribosomes. They are surrounded by membranes that record their ancient incorporation. This idea had early roots in the work of Konstantin Merezhkovsky and Ivan Vallin, but it was Lynn Margulis who forcefully revived and defended endosymbiotic theory in the 20th century. At first, many scientists resisted it. The idea that parts of our cells came from once independent bacteria sounded radical. But the evidence grew stronger. And today, endosymbiosis is one of the most important ideas in evolutionary biology. The eukaryotic cell was not built by one lineage alone. It was a chimera, a biological union, an evolutionary alliance between ancient branches of life. Possible eukaryotic fossils may go back more than 1.8 or even 2.0 billion years, but some are difficult to identify with certainty. By around 1.65 billion years ago, unambiguous eukaryotic fossils were present. The rise of eukaryotes opened new evolutionary doors. Cells could grow larger. They could organize internal systems. They could manage more complex genomes. They could develop new forms of movement. They could engulf food. They could become predators at the microbial scale. They could eventually build multicellular bodies. The world of life was becoming more complex from the inside out. Section 7. Sex and genetic variation. Evolution works through variation. Without variation, natural selection has little to act upon. For early life, variation came from mutation, gene exchange, and reproduction. But eventually, eukaryotic life developed one of the most powerful engines of variation: sexual reproduction. Sexual reproduction is not simply mating. At its deepest level, it involves meiosis, recombination, and the mixing of genetic material from different cells or organisms. It breaks and reshuffles genetic combinations. It creates offspring that are not exact copies of their parents. It brings mutations into new arrangements. It can separate harmful changes from beneficial ones. It gives natural selection more combinations to test. Sex is costly. It requires energy. It often requires finding partners. It passes only part of an individual's genetic material to offspring. Many organisms reproduce asexually and succeed brilliantly. But sexual reproduction gave many eukaryotic lineages a new evolutionary tool, a way to generate diversity, a way to respond to changing environments, a way to explore genetic possibility. The exact origin of sex is difficult to see in the fossil record because its earliest mechanisms were microscopic. The process probably evolved before scientists can recognize it directly, but by around 1.05 billion years ago, the red alga Bangiomorpha provides some of the oldest convincing fossil evidence of sexual reproduction. Its fossils show differentiated reproductive structures interpreted as spores and gametes. This means that by this point life had crossed another threshold. Cells were not only dividing, they were exchanging and recombining inheritance in more complex ways. The future of evolution became richer, more variable, more unpredictable. Section 8. Multicellular life expands. For most of Earth history, life was microscopic, but eventually some cells began staying together, not as loose colonies only, as bodies. Multicellularity evolved more than once. It appeared independently in animals, plants, fungi, red algae, green algae, brown algae, slime molds, and even some bacterial groups. This means multicellularity was not a single invention. It was a repeated evolutionary solution. When cells cooperate, new possibilities emerge. Some cells can specialize, some can protect, some can move, some can feed, some can reproduce, some can form surfaces, some can form internal structures. A body becomes more than a collection of cells. It becomes an organized system. Multicellular eukaryotes existed long before the dramatic expansion of large life near the end of the Precambrian. Fossils such as Chinchonaya, around 1.635 billion years old, have been interpreted as multicellular eukaryotes. Other fossils around 1.56 billion years old show macroscopic eukaryotic organisms several centimeters long. Life had begun experimenting with size and organization far earlier than many once imagined. But between roughly 800 and 600 million years ago, something changed. Multicellular eukaryotes became more diverse, larger, more ecologically important. The oceans began filling with more complex forms of life. Several forces may have contributed. Oxygen levels changed. Nutrients became more available. Predation increased. Cells evolved better adhesion and communication. Gene regulatory systems allowed more controlled development. Environmental disruption from severe ice ages may have opened new ecological possibilities. This was not evolution marching toward humans. It was branching, experimenting, testing forms. Some lineages would survive, some would disappear forever, but life was no longer only microbial mats and single cells. The stage was being prepared for animals. Section 9. The Ediacaran world. After the severe cryogenian ice ages, Earth entered the Ediacaran period. From about 635 to 538.8 million years ago, the oceans became home to strange and remarkable organisms. Some were large, some were soft-bodied, some looked like fronds, some looked quilted, some looked segmented, some looked nothing like anything alive today. Around 575 million years ago, the iconic Ediacara biota became prominent. These organisms spread through marine environments and left impressions in ancient sediments. They were among the first large complex life forms visible to the naked eye. Charnia rose like a branching frond. Rangea formed quilted leaf-like shapes. Dickinsonia moved across ancient seafloors, its body divided into repeated segments or quilted units. Kimberella may have grazed on microbial surfaces and may represent an early bilaterian animal. Tribrachidium displayed a strange three-part radial organization. Claudina built mineralized tubes. Namacalathus produced skeleton-like structures. Not all Ediacaran organisms fit neatly into modern categories. Some were probably early animals. Others may represent extinct experiments in multicellular life, branches of evolution that flourished for a time and then vanished. The Ediacaran world was not empty. It was not a waiting room before real life began. It was a living marine world with its own ecosystems, strategies, and forms. Organisms fed. Some moved, some grazed, some filtered particles from seawater. Some built mineralized structures. Some began disturbing the sediment. The seafloor itself changed. Microbial mats that once covered wide areas were increasingly grazed, burrowed, and disrupted. Ecosystems became more active, more physical, more interactive. Life was beginning to engineer its own environment. The Ediacaran was not the final destination. It was a threshold, the quiet before a much louder biological storm. Section 10. The Cambrian radiation. Around 538.8 million years ago, the Cambrian period began. and life entered one of the most dramatic evolutionary radiations in Earth history. This is often called the Cambrian explosion. But the name can mislead. It was not instantaneous. Animals did not appear from nowhere in a single moment. The roots of the Cambrian reached back into the Ediacaran. Life had already been experimenting with multicellularity, movement, feeding, skeletons, and body organization. But in the Cambrian, the fossil record changed dramatically. Animals became more abundant, more diverse, more mobile, more armored, more predatory, more recognizable. Mineralized shells and exoskeletons became widespread. Eyes and sensory systems evolved. Jointed limbs and specialized appendages appeared. Burrowing transformed seafloor sediments. Food webs became more complex. Predators pushed prey to evolve defenses. Prey pushed predators to evolve better tools for hunting. The oceans became an evolutionary battlefield. Armor, speed, senses, jaws, claws, burrows, shells, spines, movement. The Cambrian was the age when animal ecosystems became far more active and complex. Why does the Cambrian fossil record seem so sudden? Several forces came together. Shells and skeletons fossilized more easily than soft bodies. Oxygen levels may have supported more active metabolisms. Predation created pressure for armor, mobility, and sensory organs. Developmental genes allowed more elaborate body plans. Nutrient changes may have increased biological productivity. Burrowing and feeding changed the seafloor. Exceptional fossil sites preserved soft-bodied animals that ordinary rocks would never reveal. No single cause explains it all. The Cambrian radiation was a convergence of biology, chemistry, ecology, and environment. Charles Darwin understood how important this problem was. In his time, the sudden appearance of complex Cambrian animals seemed like a challenge to gradual evolution. Darwin argued that the older fossil record was incomplete. Later discoveries proved that life existed long before the Cambrian. Microbes, eukaryotes, multicellular organisms, Ediacaran life. The Cambrian was not the beginning of life. It was the moment animal life became far more visible, diverse, and ecologically dominant in the fossil record. Much later, Stephen Jay Gould would bring Cambrian life to a wide audience through his interpretation of the Burgess Shale. He emphasized contingency, the idea that evolution is not guaranteed to produce the same outcome if replayed. Life's history is shaped by survival, extinction, chance, environment, and opportunity. Some Cambrian forms survived and gave rise to later lineages. Others disappeared. The world we know was not inevitable. It was one path among many possible paths. Section 11. The Great Chain of Biological Revolutions. From 3.8 billion years ago to the dawn of the Cambrian, Earth changed again and again. This was not one event. It was a chain of revolutions: chemical evolution, microbial life, photosynthesis, oxygenation, eukaryotic cells, endosymbiosis, sexual reproduction, multicellularity, Ediacaran ecosystems, Cambrian animal radiation. Each revolution changed what life could become. Microbes turned chemistry into biology. Photosynthesis turned sunlight into living energy. Cyanobacteria released oxygen and transformed the planet. Oxygen made new metabolisms possible. Endosymbiosis created complex cells. Sex increased genetic variation. Multicellularity allowed bodies. Ediacaran organisms explored large life. Cambrian animals transformed ecosystems. But this was not a straight march toward humans. evolution does not move toward a final goal. It branches, it tests, it loses, it repeats. It creates forms that vanish and forms that endure. Microbes were not replaced by complex life. They remained the foundation of the biosphere. They still are. Every forest, ocean, animal, and human depends on microbial life. The story of life is not a ladder. It is a branching tree, a network of experiments across deep time. The earlier cells did not know they were the beginning of animals. Cyanobacteria did not know they were changing the atmosphere. The first eukaryotes did not know they carried the future of complex life inside them. The first multicellular organisms did not know that one day eyes, limbs, shells, brains, and civilizations would follow. They simply lived. They survived. They reproduced. They changed. And over billions of years, those small changes reshaped Earth. Section 12, Closing, From Chemistry to Animals. For almost 3 billion years, life was mostly invisible to the naked eye. It lived in mats, sediments, vents, ponds, oceans, and microscopic worlds. It left traces in stone, chemical signals, layered structures, tiny fossils, ancient carbon, mineral patterns. But slowly, life transformed the planet that held it. It changed the air. It changed the oceans. It changed the rocks. It changed itself. From non-living chemistry came the first biological systems. From microbes came photosynthesis. From photosynthesis came oxygen. From oxygen came new energy. From ancient partnerships came complex cells. From genetic recombination came new variation. From cellular cooperation came bodies. From bodies came ecosystems. From ecosystems came animals. And with animals, Earth entered a new chapter, a world of movement, predation, defense, senses, skeletons, burrowing, feeding, competition, and complexity. By the beginning of the Cambrian, life had become impossible to ignore. The planet was no longer just inhabited, it was alive in motion. The oceans were changing. The seafloor was changing. Evolution was accelerating in visible form. The long age of microbial Earth had not ended, but it had opened the door to something new. The rise of animals. The rise of complex ecosystems. The beginning of the world that would eventually lead to fish, forests, reptiles, mammals, primates, humans, science, memory, and the telling of this story. This was the origin and evolution of life. Not a single spark, not one simple beginning, but billions of years of chemistry, survival, transformation, and chance. A planet becoming alive, a biosphere learning to breathe, a living world preparing for the next chapter.